It's a real pleasure to be here for this webinar for the journal to talk about my work. So the title of my talk is Quantitative Approaches to Understand the Role of Zinc and Magnesium Dyshomeostasis on Thrombotic Risk. I was initially only going to talk about the zinc work, but I've decided to show you some of our recent magnesium data as well, which is quite interesting. So as Marco says, I'm based at the University of St Andrews in Scotland. St Andrews is quite a small place. So Marco knows this because he's visited St Andrews before. It's a town of about 20,000 people, which is completely kind of overtaken by the university. The University of St Andrews is quite an old university. It's existed for over 600 years. It's the oldest university in Scotland and the third oldest in the English-speaking world. The other thing St Andrews is famous for is golf. So it's home of the Old Course, and it also hosts major golf tournaments every now and again. So the town's recently frequented by professional golfers such as Tiger Woods and co. So it's a kind of a strange place. It's like a picture postcard picture of Scotland, located in the northeast of Fife, just by the sea. So anyway, onto the work. So I thought I would start with a slide talking about my research interests, just to summarize what we work on. So a long-term interest of mine is circulatory metal transport and handling. So that concerns how metals are carried in the body and how they're taken up by cells. We've done a lot of work on zinc, and we are interested in how zinc interacts with plasma proteins. So key ones that we've worked on are serum albumin, which I'll talk about, and another protein we've done quite a bit of work on called histidine-rich glycoprotein. In our work, we use structural and biophysical approaches. So these have included collaborating with other researchers to do X-ray crystallography. More recently, we are involved in some cryo-EM work. But we also take the approach quite widely. So we've used biophysical approaches to gain information on structure. So we've used things like HDX, MS, and also pulsed EPR-based approaches in collaboration with other experts. I'm based in the School of Medicine in St Andrews, so I'm clearly interested in disease. So we're interested in how abnormal metal handling or deficiencies in metals can lead to disease mechanisms. Particularly interested in thrombosis and haemostasis, which I'll talk quite a lot about in this webinar. But we also have some work ongoing, which involves collaboration with Marco, looking at insulin decomplexation as well. Another interest of mine is the application of omics technologies in disease, and I will talk a little bit about this. So we have an interest in quantitative proteomics and its application to better understand complications associated with metabolic disease, but we also have some projects looking at cancer as well. We are also interested in lipids and how they impact on metal biology and metabolic disease. And I'll also talk a little bit about some of our early work looking at metalloproteomics, which is some work that we're doing in collaboration with Claudia Blinder's lab at the University of Warwick in England. So first off, I'm going to talk a little bit about hemostasis, and particularly zinc's roles in hemostasis. So zinc is an essential micronutrient required by the body, and it plays important roles in numerous aspects of hemostasis. So when cells are injured, or when platelets, which are small red cells in our blood, become activated, zinc is released, and that zinc acts a bit like a signaling molecule. Now, the zinc can act both intracellularly within platelets, or it can be released by platelets and mediate processes such as activation and aggregation. So zinc is important both inside cells and on the outsides of the cells. Zinc is also important for various aspects of the coagulation cascade. So I'm not going to go through this in detail, but zinc both stimulates some parts of the cascade and inhibits other parts. So it's both an important positive and negative regulator of coagulation. And once blood clots form, zinc also plays a role in regulating the stability of the clot and the breakdown of the clot, which is a process called fibrinolysis, so the breakdown of fibrin clots. So zinc plays a role in multiple aspects of haemostasis. But its role in the blood doesn't go unchecked. So the availability of zinc is controlled by a protein called serum albumin. Now, serum albumin acts a bit like a buffer. So it carries zinc around the body, but it also acts a bit like a buffer, which controls the levels of free or available zinc. Now, in humans, zinc is present in the blood at a total concentration of around 15 micromolar, which is much lower than the concentration of albumin, which is thought to be more like 600 micromolar. So under normal conditions, you wouldn't expect any more than 1 in 30 albumin molecules to be carrying zinc. But about 75% of zinc in the blood is bound to albumin. It has a KD of around 2 micromolar. So it's kind of well optimized for the concentration of zinc which is around. But of this 3/4 of zinc bound to albumin, this represents around 98% of the exchangeable pool. So what that means is that the other 25% of zinc is bound to low abundance proteins, but bound to them generally quite tightly, and that zinc is not available. It's this pool of zinc which is bound to albumin which makes up the major fraction of what zinc is exchangeable. Now, albumin binds zinc primarily through 2 binding sites. Well, the human protein, certainly, there's 2 binding sites. There's a primary site, which is of key physiological relevance, and a secondary site, which is of less physiological relevance. The primary site binds zinc with an affinity about 10 times higher than the secondary site. The primary site is quite interesting because it's affected by fatty acid binding. So albumin doesn't just carry zinc, it carries a lot of other molecules. So these include drugs, hormones, but fatty acids are a key molecule that it carries around. Fatty acids are interesting because the levels are dynamic in the body, so they can change at different times of the day. The levels can go up if you eat food or if you exercise, but they can also be chronically elevated in some disease states, such as type 2 diabetes or obesity. So the level of fat, because they can affect binding of zinc at the primary site, there's a potential for some regulation of zinc binding there, or zinc's ability to buffer zinc levels in the blood. So this is a slide here showing the locations of the binding sites that I talked about. So on the left, you have the crystal structure of albumin. It's got 3, it's a single chain protein, but has 3 homologous domains, which are shown here in green is domain 1, yellow is domain 2, and then in this kind of bluey purpley color, we've got domain 3. But what I'm showing here is 7 different fatty acid binding sites. So you can see 7 different fatty acid molecules here bound in this structure. And they're color coded. So the ones shown in red are the fats bound to the higher affinity sites, and the blue ones are the ones bound to the lower affinity sites. Now, in the structure on the right, we've got a crystal structure that we published together with Vladek Miner's group back in 2016, and it shows the location of the primary zinc site on albumin. And you can see the zinc here shown by this purple sphere, and it binds at a site located at the interface between domains 1, shown in green, and domains 2, shown in yellow. And something that's quite important to note is that this site is actually quite close to one of the high affinity fatty acid binding sites, this FA2 site, which you can see on the left here. Now, something that we're quite interested in is this allosteric relationship between fatty acid binding at the FA2 site and zinc binding by albumin. And what I'm showing you here is an overlay of the 2 structures that were on the preceding slide. And in the zinc bound structure, you can see we've got 3 amino acid residues which come together to form this tetrahedral zinc binding site. The 4th ligand is provided by a water molecule. Now, in the presence of fatty acids, what happens is when the fatty acid binds here at this domain 1-2 interface, it causes a shift in the structure of domain 1 in relation to domain 2, such that one of the key ligands, this histidine side chain, moves around 7 to 8 angstroms away from the other coordinating residues. And this prevents zinc from binding. So what you can see on the right here is an ITC experiment, whereby we've loaded albumin with 0 to 5 mole equivalents of myristic acid here. So we've used myristic acid, which is the same fatty acid which is shown in the structure here on the left. And what happens is, as we load it with fatty acid, zinc binding is perturbed. So this is isothermal titration calorimetry, so it works a bit like a thermometer. As you serially inject zinc into the protein, what happens is you get an energy change associated with that binding. When you have no fats present, you get a high degree of binding, so a high energy change, but you see the energy change becomes less and less as you load it with fat. And what happens is, as you start to saturate the binding site or sites with the zinc, you get less binding on incremental injections. So that's what we see here on the right. So the upshot is that fat loading of albumin prevents zinc binding with high affinity. So recently we undertook some work to show that it was the FA2 site that was responsible for this interaction. So we generated a triple mutant form of albumin which lacked the FA2 site. So this is the triple mutation that we made. So there are 3 key residues here which form hydrogen bonds with the carboxylate group of the fatty acid. So this has these hydrogen bonds here which dock the fat in here, and then there's a binding cleft which is lined with hydrophobic amino acids. So what we wanted to do was remove these hydrogen bonds, or the potential to make these hydrogen bonds, to reduce the affinity of the fatty acid binding at that site, and perhaps push fatty acids to binding at the other sites on the molecule. So we mutated these 3 residues, and what you can see on the bottom here, so this is some carbon-13 NMR, and what we've used here is carbon-13 labeled palmitate. And so what happens is here is that when you bind the palmitate, you get 2, so this is in the presence of 1 mole equivalent of palmitate, you get 2 peaks in wild type albumin, a large peak here around 182 ppm, and then one just slightly downfield, there's another smaller peak. Now, it's been shown previously that this larger peak is binding at the FA5 site, and the smaller peak is binding at the FA2 site. But what you can see is in our FA2 knockout, our triple mutant, we don't get that little peak. So we've removed binding at FA2. And then what you see is another experiment on the right here. So this is ITC again, but this time we're looking at myristate. We've used myristate fatty acid to look at myristate binding. And you can see that in the triple mutant, there's less binding than there is in the wild type protein. And that's because we think we've removed one of the sites by making this triple mutant. So you still get binding at the other sites, but at this FA2 site, there's no binding. So we think that that's what this energy change relates to, the loss of that site. So what we did was we kind of repeated the experiment that I showed you a minute ago, where we loaded albumin with different concentrations of fatty acids. This time we used palmitic acid. So in the presence of 0 to 5 mole equivalents of palmitic acid, you get this perturbation in zinc binding, which we saw before with myristate. But in the triple mutant, it makes it completely resistant to the fatty acid. So zinc is still able to bind as well, pretty much even in the presence of 5 mole equivalents of palmitate, which was not the case with a wild type protein. And what this suggests is that the FA2 site in itself is responsible for this reduction in zinc binding by albumin. So I just wanted to highlight another study that we published back in 2019. So this was an experiment that was actually carried out by my collaborator, Claudia Blindauer. So what we did here was we took blood plasma and we added myristate to a batch of the blood plasma, and we compared the non-myristate with the myristate added plasma. So what we did was we separated each of these plasma preps out using size exclusion chromatography. So we got a range of different fractions, And the black line represents the kind of ACTA trace, which shows the protein coming off the column. And it comes off in two large fractions, basically. You get two large peaks. Now, if we take each of the fractions and we measure how much zinc is in them, what you see is that in the absence of myristate being added, most of the zinc is associated with the proteins which come out in the second peak, not so much with the first peak. However, when the plasma has already been spiked with myristate, we get similar degree of separation. So we're not affecting protein separation. That looks like it's following largely the same profile, but we do see a change in where the zinc is. The plot on the right here just shows that as we go through the fractions, if we add on all the zinc that we're able to detect, it just shows that we're able to get all the zinc back. So we're not losing zinc somehow. It's basically the zinc is moving from some fractions to other fractions. If we probe the fractions by Western blotting, what you see is the fractions associated with the second peak is where the albumin is. So what we think we're doing here is that in the normal plasma, we see most of the zinc bound to the albumin, but when we spike with myristic acid, we get less binding to albumin and more zinc association with other proteins. So this is quite important for albumin's function as a zinc buffering protein. If fatty acid levels are high, its ability to buffer zinc, so if we were reacting to zinc being released from a cell, for example, you would maybe see less albumin binding and more binding to zinc responsive proteins. So you could end up with perhaps an exaggerated response. So we were interested in whether this process might influence fibrin clotting. So thrombosis and aberrant haemostasis is associated with diseases whereby you have high levels of fatty acids. So we're talking about obesity, type 2 diabetes, fatty liver disease, some types of cancer. You also have high levels of fats in the blood, and you also have an increased risk of thrombosis. So wherever fats are high, there's an increased thrombosis risk. And we were interested as to whether this mechanism may be responsible. So to begin to look at this, we had to look at fibrin clot formation. So the schematic diagram on the left shows the polymerization of fibrinogen to form fibrin clots. So fibrinogen is a triple chain molecule, and what happens is after cleavage by thrombin, it starts to aggregate in a lateral way to form these protofibrils. Further cleavage from thrombin can then cause these protofibrils to come together and form lateral aggregation, with most of the aggregation being controlled by the alpha domain and also the gamma domain. So you see a lot of these protein-protein contacts in these fibers are being instigated by alpha and gamma domains of the protein. Now, this is a process that we can follow using quite a simple assay. So if we take plasma or we take purified fibrinogen and we add thrombin, we would also need calcium to be present. What happens is that you start to get an increase in absorbance as the fibrinogen comes out. It forms fibrin and the fibrin aggregates and it comes out of solution. So what happens is you get an opacity or an increase in optical density caused by the formation of the clot. Now, in a plasma sample, you can get quite a lot of information from that curve. So, for example, the maximum absorbance, the absorbance that it stops at, gives you some information about the size and the density of the clot that forms. You can also measure the rate, which will tell you about the propensity for that clot to form. There's also a lag time, which can tell you a little bit about how easy the clot is to form as well. But the other thing you can look at as well is clot breakdown or clot lysis. So for that, if you add tissue plasminogen activator along with plasminogen, what you can then do is then break down the clot, and the reverse process happens where you get a reduction in the turbidity or opacity as the clot breaks down and it returns to rest. Now you can get an idea of how easy that process is to occur by looking at the rate, or often a key measurement is the time to get to 50% lysis, which is shown by E here. So you can get quite a lot of information about how blood clots form and how they break down just using a simple turbidimetric assay. So we started to do these assays and get an idea of what happens when you add zinc and fatty acids. So if you take pooled citrated plasma and you recalcify it and add thrombin, a clot will form, and it has a characteristic maximum absorbance, which you can measure. If we add, here we've added myristic acids, we add a fatty acid here, what we see is we've got an increase in the amount in the maximum absorbance, suggesting that we're forming a larger or denser clot when myristate is present. If we do the same experiment, but we add zinc, what you see is that the zinc, even in the absence of added myristate, increases the size and density of the clot. And then when we add myristate, that's exaggerated even more. So that suggests that the zinc in the myristate acts synergistically to increase this maximum absorbance related to clot size and density. So that's quite interesting. After this, we then decided to look at patients. So we managed to get plasma from individuals with type 2 diabetes and also some age-matched controls. And what we found was that the free fatty acid concentration, as expected, was higher in the type 2 diabetes patients than it was in the healthy controls. And similarly, when we formed clots, we found that the maximum absorbance was higher in the type 2s than it was in the controls as well. We looked at albumin concentration, and actually it was higher in the type 2s than it was in the controls, suggesting that they should be able to buffer their zinc better because they have a bit more albumin. When we looked at zinc, there was no statistically significant difference between the controls or the type 2 diabetes. But the key thing here is that fat levels are higher and maximum absorbance is higher. We wanted to learn a little bit about what zinc was doing to the structure of the clot. So we did some experiments using scanning electron microscopy to actually image the clot fibers that form. And what we did was we measured the diameter of the fibers in the clots under different conditions. So in the experiment on the left here, what we did was we used a purified system where we just took pure fibrinogen, but we also added serum albumin. And what we did then was we formed the clot by addition of thrombin and calcium. And what we saw was that in the presence of zinc, the average fiber diameter got larger. So the fibers got thicker when we added zinc in this purified system. We then decided to look at the patient samples and measure the fiber diameter. And what we saw was that in the controls, we had a certain fiber thickness, but it was higher in the people with type 2 diabetes. When we added zinc, we again saw that the fiber thickness was higher than it was in the controls without adding zinc. And similarly, again, we got an increase in the type 2 diabetes when we added zinc, again suggesting that the zinc somehow increases the fiber thickness, and that in the people with diabetes, they were the zinc seemed to have even greater effect, potentially caused by the elevated levels of fatty acids, reducing the albumin in their plasma from buffering the zinc. So more zinc is binding to the fibrin and causing this change in clot structure. We then did a lipidomic analysis to see what fatty acids were present in the samples. And so this was the results of that experiment. So you can see that in the type 2 diabetes plasma samples, there was a much higher concentration of a number of different fatty acids, which you can see here compared to the controls. What we then did was we took the individual fatty acid levels and we looked to see if there was any relationship with this increase in maximum absorbance we saw in our turbidity experiments. So this is the data that we got. So this is looking at associations between along the bottom on each of these axes, we have maximum absorbance, and then on the vertical axis, we've got the individual fatty acid concentration. So what we saw were relationships between all of the saturated fatty acids, so myristate, palmitate, and stearate, and we also saw significant correlations with two unsaturated fatty acids, which were oleic acid and vaccinic acid as well. So again, this points towards that potential relationship between fatty acids and zinc all caused through aberrant buffering of zinc by serum albumin. So how does zinc impact upon the fibrin network? So clearly there are direct effects. So it's been known since the 1980s at least that zinc binds to fibrinogen and fibrin. This is a paper from back then by Gerard Marx. In this paper, he found that fibrinogen has multiple zinc binding sites and an average KD of around 8 micromolar, which is actually quite comparable with serum albumin. It's only a little bit higher. So you would think that fibrin would be well positioned to pick up zinc that albumin was unable to bind. It seems to have an affinity which is in the right ballpark range. We also more recently have started to look at where putative zinc binding sites on fibrinogen might exist. So they've been located so far to the alpha, C and gamma domains, which are quite important parts for fibrin clot formation, but specific residues involved in these sites have not been identified. There is a partial structure of fibrinogen in the PDB, and it shows parts of the alpha chain as well as the beta chain and gamma chain. We've located a potential site to the gamma chain right on the bulb bit, which can interact with other molecules, other fibrinogen molecules. And you can see this putative site here, which looks a little bit like the zinc site on albumin, where we have 2 histidines and an aspartate here. What's quite interesting is this aspartate has been known to be mutated to another residue in some individuals, and those individuals have problems with bleeding. They find it difficult to form blood clots. So we think this could be a putative zinc site, which is important for regulating fibrinogen or fibrin interactions during clot formation. Unfortunately, the alpha C domain is not present in the available structure, but based on the amino acid sequence, there are a couple of motifs which are known to bind zinc in other proteins. So there's this HXH motif located here in the sequence, and another HEXXH motif, which is located here. So we've currently got funding from the British Heart Foundation to make mutations in fibrinogen and to look to see if this affects zinc binding, and then again what the knock-on effects of these mutations might be on clot structure and clot formation. So that's some ongoing work we have at the moment. So I just want to show you this is unpublished data. It's kind of preliminary work, but I just wanted to show you some of the things we're trying to do to try to work out what's happening to the zinc that's not binding to albumin. So what proteins pick up the zinc? We showed previously in that metabolics paper that the ability of albumin to bind zinc changes when you add fatty acids, but what proteins actually pick up the zinc? So that's something that we've set about trying to identify. We know that fibrin might bind some of the zinc, but fibrin doesn't normally exist in plasma. It's only existing when it's cleaved by thrombin. What other proteins present there are picking up the zinc? So here we did an experiment whereby we separated the plasma a bit like before. So we've got a normal plasma with no fatty acid added, and a set of plasma where we've added 3 millimolar myristate. But instead of just separating the plasma by size exclusion, we've separated the plasma in 2 dimensions using ion exchange in one dimension and size exclusion in the other. And then to each of these fractions, we measured where zinc was present. So this work was carried out by Claudia Blinder's lab. And what you can see is most of the zinc is associated with this red part where a lot of protein is present. And what we think is that this red protein corresponds to serum albumin, which is known to be the most abundant protein in the blood. So we think this red patch is mostly albumin. And then you can sort of see zinc bound here. In the fat spiked one, you can see we get a similar profile of separation. And the zinc, again, it does look like there's a lot associated with the albumin. But when we take a difference of the 2 together, we see that some of the zinc has moved from some of the fractions into other fractions. So the blue fractions are losing zinc, the yellow fractions are picking up zinc. So what we wanted to do is see if we could identify which proteins were picking up the zinc. So to do that, Claudia sent the samples to me, And then here in St Andrews, what we did was LCMSMS. And what we did was we then sort of profiled each of the fractions to see what proteins were present. And we also tried to get an estimate of the abundance of each of the proteins using a measure called the exponentially modified protein abundance index, or EMPAI. Now, across our fraction set, we were able to identify 716 different plasma proteins. And what we did was we looked for correlations between this EMPAI for each of the proteins and the metal concentrations present in those fractions. So this is the data that we got for zinc. So as I said, we've not published this yet. It's quite preliminary. There's a few things that we're still working on here, but I just wanted to share with you our preliminary findings. So in the absence of fatty acid, we've not added any fatty acid. It's just endogenous plasma. Albumin was the main protein found to be associated with the zinc, which was as expected. We also saw significant correlations with a couple of other proteins, including alpha-1 antitrypsin and antithrombin 3. We saw significant correlations at the p-value level, also with alpha-1b glycoprotein and a protein called gelsolin. Now, in the myristate spike plasma, we saw a bit of a difference. So now the primary protein that seems to be associated with zinc is this protein kininogen, and it has a very low p-value. Interestingly, albumin is also on the list, but the significance has gone down somewhat, and it's now fourth on the list compared to having a very, very low adjusted p-value in the non-spiked plasma. So this highlights that albumin is losing zinc, but also it's providing some evidence that kininogen is perhaps picking up some of the zinc released by the albumin. Now, kininogen is quite an interesting protein, and it's involved in haemostasis. So high molecular weight kininogen is known to be a key modulator of the intrinsic pathway, and it's involved in switching on the intrinsic pathway of coagulation. So it interacts with other molecules, and it's known that zinc helps kininogen to interact with these molecules. And the zinc generally has a prothrombotic effect on kininogen, so can help to initiate clot formation through this intrinsic coagulation pathway. So currently in the lab, we're looking at how zinc influences kininogen functioning using a range of functional assays. And we're also looking at the impact of fatty acids on that process as well. So this is work that we're undertaking at the moment, which is ongoing. Just to go back to the metalloproteomic work, so just to say that as well as zinc, we also looked at a couple of other metals. So we looked at copper and also iron to try and identify what the copper and iron speciation might be using our approach. Copper seemed to be involved with the fractions you can see indicated here in the top right, and iron associated with fractions you can see down here on the bottom right. When we looked at associations with our EMPI values across our proteomic data, this is the data for copper. And what was quite pleasing to see is that ceruloplasmin, which is the main copper binding protein in the blood, came out as the most significant hit. We also saw a couple of other proteins, including alpha-1 anti-chymotrypsin, which had a significant adjusted p-value, and a range of other potential copper binding proteins, including kininogen actually, which have p-values in the significant range. Unfortunately, due to this multidimensionality of the data, they did not survive multiple hypothesis correction, but they still have adjusted p-values below 0.2. This is the list for iron, and pleasingly again, transferrin came out as the main protein associated with iron with a very, very significant adjusted p-value. We identified a few other potential iron binding proteins, including hemopexin, which is known to be a heme scavenger, so you would expect it to be associated with iron as well. So at this point in the webinar, where we're looking at other metals in the blood, I think this is a good time to make a segue into another area of work that we are interested in, and that concerns the metal magnesium. So this is some data from a study that we published a few years ago, where we had a type 1 diabetes cohort and matched controls. And what we found was that when we looked at the plasma metal levels in these samples, we found that the type 1s were slightly deficient in magnesium compared to the controls, pointing towards people with diabetes having less magnesium on average than healthy people without diabetes. So we were interested as to whether this magnesium had any effect on haemostasis. So we did some clot structure, some turbidity experiments to look at how the magnesium affects the size and density of the clots. And what we found was that the maximum absorbance didn't change. So the size and density of the clots, it would suggest that they're similar in type 1 diabetes compared to controls. However, we did see a change in the lysis time. So this is the time to 50% lysis after adding plasminogen and TPA. And what we found was that it took a little bit longer for the clots to break down in type 1 diabetes compared to controls. And this sort of delay in clot lysis is referred to as hypofibrinolysis, and it's a well-known feature in type 1 diabetes, hypofibrinolysis. And it's known to lead to thrombotic effects in those patients. This difficulty that the patients have, or more difficult to break down clots. This wasn't something that differed between males and females, so we did look to see if there was a sex-dependent effect. So there wasn't there, but there was this effect on lysis time. So then we looked to see, we know we've got a range of magnesium concentrations in this cohort. Is there a relationship between lysis time and magnesium concentration? And we found that there was. And actually the patients that had very low magnesium concentrations actually had very long lysis times. So we tended to see it's significant linear relationship, but actually it looks like this might not be a fully linear relationship. And actually there could be there could be an even steeper line if we took, for example, the very low levels of magnesium. So that might suggest that magnesium is somehow having an effect on clot formation. As I pointed out before, there wasn't an effect on maximum absorbance. And again, when we looked at fibrin fiber diameter, we didn't see any significant difference in the clot structure. We think it's more to do with the enzymes involved in breaking down the clot once they form. More recently, we managed to get hold of some data which involved medical health records from Scottish patients. So we looked in a very large cohort of participants. And so here we have over 120,000 people. So we looked across those people to see if they had high, normal, or low levels of magnesium. So these are people who had a magnesium test done. And we compared the effects of having a low level to a normal level and the future development of different cardiovascular diseases. And what we found was that low magnesium seemed to be a risk factor for hypertension and also a whole range of vascular diseases as well. Interestingly, it was slightly protective for ischemic heart disease and pulmonary heart disease. But what it suggests is that maintaining a normal magnesium level is quite important, particularly in the context of vascular disease. So this is some recent data that we published this year. So at the moment, we're interested in the prospect of using magnesium supplementation as a way to perhaps reduce the number of thrombotic complications that happen in people with type 1 diabetes. So we have some Diabetes UK funding at the moment to undertake a supplementation trial. So this is work that we're doing in collaboration with Ramsey Ajan's group at the University of Leeds. So what we're doing in the trial is we're screening potential participants to see what their magnesium levels are like. For those that have a low level, we are then recruiting them into the trial. And then the trial has a crossover structure whereby half the participants will be given a high dose magnesium for 2 weeks, half will be given a placebo. We'll then have a 2-week washout, and then the participants will swap swap over. So the placebo ones will be given the high-dose magnesium, and the high-dose ones will be given the placebo. We will take plasma samples at various stages, and then we're going to look at fibrin, clot, and lysis properties in these participants. So we're going to see if this works as a way to reduce the thrombotic burden, which can lead to a whole range of different disease states in these patients, and costs health services, billions of pounds a year. Whereas magnesium is quite a cheap treatment, and it doesn't require prescription. So this could be quite a nice way to reduce the burden if it works. So this is ongoing work at the moment. So just for the last little part, I'm not going to spend too much time on this, but I'd like to talk a little bit about some of our quantitative proteomic work. So we've published a number of papers using a technique called swath MS to undertake quantitative proteomics in different sample sets. So we published a paper a few years ago looking at hydroxyapatite binding proteins in age-related macular degeneration patients. So that interaction is particularly important for the pathology of that disease. We also looked at proteomic changes in dendritic cells during activation by lipopolysaccharide. We've also published a paper looking for prognostic markers for esophageal cancer using a sample set. These were from esophageal cancer patients. We got the samples from Dublin. They were recruited as part of a study there. And then finally, we published a paper. We played quite a small part in this paper together with Mark Malloy and Alicia Jenkins' group. And so they're based at the University of Sydney in Australia. But this paper basically highlighted a workflow for large-scale proteomic analysis of a large number of samples at the same time. So basically to help enable quantitative proteomics to be applied to larger trials. More recently, we've been interested in using quantitative proteomics to look at changes in plasma proteins and what effects that might have on haemostasis. So again, as always, we're interested in metabolic disease, but we managed to get hold of a sample set where we had 25 individuals who underwent gastric bypass surgery, and we got samples from them both before and 9 months after successful surgery. So usually patients are only recruited for bariatric surgery when they reach a BMI of 40 or 35 with some medical complications. So that might be type 2 diabetes. So in the sample set, we had an average BMI before surgery. It was very high. It was 51.8. But the surgery was successful, and the patients lost a lot of weight over the 9 months after the surgery took place. And so the weight dropped from an average of 150 kilograms to 105, and the BMI dropped significantly from 51.8 to 37.2. So there was also a significant reduction in a number of other anthropomorphic measures, such as waist circumference, hip circumference, etc. These are some clinical measures. I'm not going to go through them in detail, but just to say we did look at fatty acids, and for a wide range of the fatty acids, the levels reduced after surgery. This is just a slide here looking at clot parameters again. And what we found here was that the maximum absorbance was very high preoperatively, and then it went down after the operation, suggesting that we were getting a reduced size and density of the clot in the participants after surgery and weight loss, which may be associated with, well, it's consistent with the fat levels going down and potentially zinc being better controlled. Zinc levels increased in the participants as well, but that zinc would be better buffered. You often see a sort of yin and yang effect with copper. So zinc increased, copper decreased, and the zinc copper ratio you can see was much better after the surgery. So we've also undertook some quantitative proteomics on these samples. So we did this DIAMS based proteomics. So across the sample set, we identified 332 proteins. We saw significant differences for 70 proteins, 27 went up, 43 went down after surgery. We were able to see changed proteins associated with a range of processes, but not least haemostasis and platelet functioning. So we saw changes that can help to explain some of the data that we got. So it might not all be down to zinc and fatty acids, but obviously proteomic changes happen and contribute to the sort of changes in haemostasis as well. So I think this is my last data slide. I know that the data here will be, you know, too small to read in detail. I'll take you through a few highlights. So these are proteins that changed in the data, and looking at correlations between different clinical measures, including maximum absorbance and other clot parameters, including clot lysis. But what we saw was that max absorbance and magnesium levels negatively correlated with a number of coagulatory proteins, including factor 2, factor 10, factor 12, and prekallikrein. Factor 2 is, of course, prothrombin, which forms thrombin, which cleaves the fibrinogen. The max absorbance in magnesium also correlated positively with alpha-2 macroglobulin, which is an inhibitor of some hemostatic enzymes, including thrombin, factor 10A, and also plasmin, which breaks down the clot. Many inflammatory and stress-responsive proteins also correlated with the fibrin, clotting, and lysis parameters, as well as lipid parameters, anthropomorphic measures. In our study, very few proteins actually correlated with zinc, but this possibly reflects that zinc's role is more to do with handling than actual zinc levels. So we didn't see any real associations with zinc. So anyway, I think I've spoken for long enough. So just to conclude my talk, I hope you can see that fatty acids alter zinc speciation in type 2 diabetes via an allosteric switch on serum albumin. This potentially increases the proportion of zinc bound to other proteins, which can have direct and indirect effects on the fibrin network, leading to altered fibrin clot structure and clot parameters. Plasma magnesium concentrations are lower in type 1 diabetes and correlate with an increased clot lysis time. Magnesium supplementation in type 1 diabetes may be a cheap, effective way to reduce thrombotic complications, and that's something that we're currently investigating. And finally, I hope I was able to show in the brief time afforded that quantitative proteomics is an invaluable approach for understanding metabolic disease states and haemostasis and the mechanisms that increase the risk of thrombotic complications. So I'd just like to leave it there by thanking a lot of people who were involved in various aspects of what I presented, particularly my research group are shown in blue here, include my current group and some past members who've contributed to some of the data I presented. Down here are other collaborators in St Andrews who've helped with the work. I'd like to highlight key collaborators, including Marco Kettley from Marco's lab, actually spent time in my lab, which is why she put her in blue here. Also Claudia Blinder, who we've been working with for a long time on zinc handling, and Ramsey, who's probably, I would say, the key clinical collaborator that I have, who's based at the University of Leeds. And then I'd like to thank all the funders which have funded various aspects of what I've presented here. And I'd like to thank you very much for your attention as well. Thanks very much.